Welcome to this overview on drug discovery, where we'll explore how new medicines are found and developed. In this session, I'll guide you through the main stages from identifying targets to clinical trials, with real-world case studies and the challenges researchers face. Think of this as a roadmap to how science turns ideas into treatments. On this contents page, we outline the journey ahead. A high-level overview, target identification and validation, finding and optimizing lead compounds, preclinical and clinical development, the central challenges, and illustrative case studies. I'll touch on methods, computational tools, safety assessments, regulatory steps, and examples that show how discoveries translate into real patient benefits. Here we define what drug discovery really means. It's the process of finding new chemical or biological candidates that can treat specific diseases. It's inherently multidisciplinary, drawing on biology, chemistry, pharmacology, and computation, and it covers everything from basic research to molecules ready for clinical testing. The image shows a typical lab setting where these early efforts take place. Drug discovery matters for several reasons. First, it creates therapies for previously untreatable or poorly managed diseases and improves patient quality of life. Second, successful drugs can transform public health by reducing mortality and morbidity. Third, the industry drives economic growth and jobs, while advancing scientific knowledge that opens new medical directions. The history of drug discovery moves from natural remedies to modern high-tech approaches. Early medicines came from plants and minerals, while the 19th and 20th centuries introduced synthetic chemistry and antibiotics. Recent decades have seen genomics and high-throughput methods revolutionize the field, and today innovations like AI continue to push drug discovery forward in new directions. When we look for places to intervene in disease, several biological targets stand out. Receptor proteins that mediate signals, enzymes that control metabolic pathways, genetic markers that point to disease drivers, and pathway modulators that can restore normal function. Each target type offers different opportunities and challenges for designing effective and selective therapies. Identifying promising targets uses diverse methods. Genomics pinpoints genes and mutations linked to disease. Proteomics reveals relevant proteins and interactions. Phenotypic screening observes functional effects in cells or organisms, and bioinformatics integrates large datasets to prioritize targets with the most therapeutic potential. Together, these approaches narrow down where drug efforts should focus. Validating a target confirms it's worth pursuing and uses multiple techniques. RNA interference and CRISPR let researchers silence or edit genes to observe effects. Knockout animal models reveal biological roles in whole organisms, and biochemical assays test direct interactions between a drug and its target. These steps reduce risk before investing in drug development. Screening techniques help find the first chemical starting points. High-throughput screening lets automated systems test thousands of compounds quickly. Target-based assays focus on specific proteins for selectivity, and phenotypic screening measures biological effects without needing a known target, often revealing novel mechanisms visible in the accompanying lab image. Computational methods accelerate discovery by simulating and predicting interactions. Molecular docking models, how compounds fit into targets. QSAR links, chemical structure to activity, to guide optimization. And virtual screening searches, large libraries to prioritize candidates for lab testing. The central graphic highlights how computation and structural data inform experimental decisions. Natural products remain a rich source of inspiration, offering bioactive metabolites from plants, microbes, and marine organisms. Ethnopharmacology taps traditional medicine knowledge to guide discovery, and structure modification adapts natural molecules to improve potency, safety, and drug-like properties. These approaches bridge biodiversity and modern medicinal chemistry. Chemical modification is a core part of lead optimization, where altering functional groups can enhance potency or specificity, improving solubility boosts, absorption, and bioavailability, and stability enhancements prevent premature degradation. The photostrip evokes the hands-on chemistry work, where molecules are tweaked to become viable drug candidates for further testing. Structure-activity relationship, or SAR, explores how molecular changes affect biological effects. Studying molecular dynamics helps understand binding interactions, optimizing receptor specificity, reduces off-target effects, and predictive modeling uses computational correlations to guide which chemical tweaks are most likely to improve activity. Visual models help make these structure-function links tangible. Evaluating toxicity and efficacy early is crucial. Dose-response testing determines therapeutic windows while avoiding harm. In vitro, screening assesses safety and activity in cultured cells before animal studies, and side effect profiling aims to identify and eliminate compounds that cause severe adverse reactions. The lab image underscores the experimental work behind these safety checks. In vitro, studies provide controlled environments to examine drug effects. Using cellular models, we can probe drug-target interactions and initial efficacy. High-throughput assays speed up testing of many candidates. Mechanism analysis reveals how a compound works at the molecular level, and predictive biomarkers help forecast later success or safety in patients. In vivo, studies move research into living organisms to assess pharmacokinetics, pharmacodynamics, and overall efficacy. Animal testing helps evaluate real-world drug behavior. Dose optimization finds safe, effective regimens. Disease modeling mimics human conditions for more accurate evaluation, and long-term studies look for prolonged effects, accumulation, or delayed responses over time. Toxicology assessments cover short and long-term safety. Acute toxicity tests identify immediate risks after high doses. Chronic studies evaluate effects of sustained exposure. Organ-specific assessments monitor potential damage to liver, kidney, or heart. And reproductive toxicity examines impacts on fertility, pregnancy, and offspring development to ensure safe use across populations. The clinical trial phases progressively test safety and effectiveness in humans. Phase I focuses on safety and dosage in a small group of healthy volunteers. Phase II evaluates efficacy and continued safety in patients with the target condition. Phase III expands comparison against standards or placebo to confirm benefit and collect extensive safety data. And Phase IV monitors long-term outcomes after approval. Ethical considerations are central at every stage. Informed consent ensures participants understand risks and procedures. Privacy protection safeguards personal data through strict confidentiality. Equitable selection aims for diverse representation to reduce bias. And risk minimization prioritizes patient safety through careful monitoring and protocols to quickly address adverse events. Regulatory guidelines guide how studies are designed, run, and reported. Approval pathways, like those overseen by regulatory agencies, demand rigorous documentation. Good clinical practice ensures ethical conduct and data integrity. Accurate drug labeling reflects clinical findings for safe use. And continuous review by regulators helps maintain public safety through inspections and oversight. Developing new drugs is expensive for several reasons. Extensive preclinical and clinical research requires major investment. High failure rates mean many projects fund few successes. And meeting strict regulatory requirements adds compliance costs for testing and documentation. The images reflect the lab work and infrastructure behind these substantial financial commitments. Drug development timelines can be long and complex. From initial discovery to market approval, the process often spans over a decade due to rigorous testing and regulatory review. Decision delays during approval or requests for more studies extend timelines. And conducting complex multi-center trials requires careful coordination and reproducibility to ensure reliable results. Drug resistance presents a growing global challenge. Microbial adaptation from overuse of antibiotics reduces treatment effectiveness. Genetic factors in cancers and other diseases can cause resistance to therapies, and an insufficient pipeline of new classes of drugs means we often lag behind evolving resistance, underscoring the need for continuous innovation and stewardship. Our first case study looks at antibiotics, where penicillin's accidental discovery sparked a revolution in treating bacterial infections. Antibiotics dramatically lowered mortality from pneumonia and tuberculosis, yet overuse has driven resistance, creating a pressing need for next-generation agents and strategies to preserve the benefits of these life-saving medicines. The second case study examines anti-cancer drugs, tracing roots from wartime chemical agents to modern targeted therapies and immunotherapies. Advances in molecular biology enable drugs that specifically target cancer-causing proteins, while immunotherapy harnesses the immune system to fight tumors, transforming outcomes for many patients and highlighting precision medicine's impact. In vaccine development, we see innovative approaches like mRNA technology that enable rapid design and scalable production, AI tools that improve antigen selection, and global partnerships that speed development and distribution. These collaborative, technology-driven advances illustrate how modern drug discovery approaches can be applied to prevent disease at a global scale. That brings us to the end of this presentation. Thanks for following along. We covered the basics of drug discovery, from targets and screening methods to validation, preclinical and clinical steps, safety, regulations, and case studies that illustrate real-world impact. If you'd like, we can dive deeper into any section or explore specific examples in more detail.